Unit 2 Viruses, Replication Structure 2.1 Introduction Objectives 2.2 Replication, General Account, 2.3 Replication of Bacteriophage, DNA virus, T phage, 2.4 Lytic Cycle Viruses, Replication 2.5 Lysogenic Cycle 2.6 Replication of RNA Virus, Tmv, 2.7 Summary 2.8 Terminal Questions 2.9 Answers Glossary 2.1 Introduction. In the previous unit you have studied that viruses cannot replicate on their own, and for replication they require machinery of the host cell for synthesis of the viral genome and proteins. As the virus, virion, cannot replicate outside the host, it has to first enter the host cell and then replicate. Viral replication results in many progenies. When these progenies leave the host cell, they repeat the cycle, infect other cells, and spread the infection. The viral nucleic acid, DNA or RNA, possesses genes, needed for synthesis of new viruses. These genes carry information and help in the formation of viruses' structural components such as capsid proteins, and few enzymes required in the virus life cycle. Some of the viral enzymes are encoded by the viral DNA/RNA, however these are synthesized and are functional only when the virus is inside the host cell. The larger viruses with DNA having more genes may also contain one or few preformed enzymes, which usually help the virus to penetrate the host cell or in some cases may help in the replication of the viral genome. The key to control viral infections lies in our understanding of details of viral replication cycle. In this unit we will study the steps involved in the process of viral replication, namely, attachment/adsorption, penetration and uncoating, biosynthesis, assembly and release. We will also discuss the replication cycle of one DNA virus, T phage, and one RNA virus, Tmv. Also in this unit emphasis will be given on the lysogenic and lytic cycle of viral replication. Objectives: after reading this unit, you will be able to know the importance and different ways in which viruses replicate, understand and differentiate between lysogenic and lytic cycle of replication, and distinguish between DNA virus, T phage, and RNA virus, Tmv, replication. 2.2 Replication, General Account. One of the most unique events in nature is the process of virus replication. The two experiments that laid the foundation of our understanding on replication in viruses are Ellis and Delbruck experiment, Hershey and Chase experiment. Ellis and Delbruck experiment in 1939. Ellis and Max Delbruck demonstrated virus replication. Their experiment is referred as single burst or one-step growth curve. To study viral replication, Ellis and Delbruck added bacteriophages, bacterioviruses, to a culture of rapidly growing bacteria. After a few minutes the mixed culture, bacteria and virus, was diluted so that interaction between phage particle and bacteria became minimum. Samples of mixed culture were taken at different time intervals and plated on lawns of bacteria covering the entire surface of the agar in a Petri plate, Fig 2.1a, for presence of phage particles. After incubation, small craters, plaque or hole in a monolayer of bacterial cells were observed on the lawn of bacteria, Fig 2.1b. At the start of the experiment, the plaque count was relatively constant for samples plated during first hour, but after that it increased hundredfold within a few minutes. During the first phase of infection, the latent period, no progeny particles were liberated from the infected cells. Then lysis of bacterial cells occurred, resulting in release of newly synthesized bacteriophages. The average number of bacteriophages released per bacterium was called the burst size. There was an increase in concentration of phage particle with time, Fig 2.1c. This was the first experiment to show the three essential phases of virus replication: I. Initiation of infection; 2. Replication and expression of virus; 3. Release of mature virus from infected cells. Hershey and Chase experiment. The second key experiment on virus replication using bacteria was performed by Alfred Hershey and Martha Chase in 1952. They worked with bacteriophage T2, which infects Escherichia coli. The bacteriophage T2 consists of an outer protein coat with DNA inside. Hershey and Chase grew cultures of E. coli in media containing radioactive S35, sulfur in sulfate form. Sulfur is an important constituent in the amino acids cysteine and methionine, which makes part of the protein coat. Fig 2.1, a. Bacterial lawn, b. Plaque-forming units in bacterial lawn, c. Graph showing an increase in concentration of phage particle with time. The single burst experiment, Ellis and Delbruck, 1939. And another set of E. coli culture in media containing radioactive P32, phosphorus in phosphate form. The element phosphorus is present in a DNA molecule but not present in any of the 20 amino acids from which proteins are made. So Hershey and Chase had one set of E. coli with radioactive S35 sulfur and another set of E. coli with radioactive P32 phosphorus. These bacterial cells with radioactive sulfur and radioactive phosphate were infected with bacteriophage T2 phages separately, Fig 2.2/2.3a. These progeny phages in E. coli cultured on media with radioactive S35 had radioactive sulfur atoms, incorporated into their protein coats. But DNA of these phages was non-radioactive. Fig 2.2. The Hershey and Chase experiment where phages were grown with radioactive S35. On the other hand, the progeny phages in E. coli cultured on media with radioactive phosphorus, P32, had P32 incorporated into the DNA. New cultures of non-radioactive E. coli were infected with S35 and P32 bacteriophage T2, respectively. After a few minutes post-infection, the infected culture was centrifuged to remove the virus coats, Fig 2.2b, 2.3b. The non-radioactive bacteria infected with bacteriophages T2 containing P32, radioactive, became radioactive and, much of the radioactivity was passed on to the next generation of bacteriophages T2, Fig 2.3c. However, in the non-radioactive bacteria infected with bacteriophage T2 labeled with S35, practically no radioactivity could be detected, as the virus coat was removed during centrifugation, in the infected cells, Fig 2.2c. Fig. 2.3, the Hershey and Chase experiment where phages were grown with radioactive P32. From these experiments, they concluded that the DNA component of the bacteriophages T2 was injected into the bacterial cell while the protein component remained outside and that DNA was the genetic material of T2 bacteriophage. The above experiments were instrumental in our understanding of viral replication. Viral replication. A virus uses life processes of host cell for synthesis of viral protein and DNA/RNA to replicate. The viral replication causes biochemical and structural changes in the host cell, which may lead to either changes in cell function or destruction of the cell. When we suffer from common cold, we are infected by the common cold virus known as rhinovirus. Some of our cells die through lysis, bursting, or apoptosis, programmed cell death or cell suicide, releasing all progeny virions at once. In some cases of virus infection, such as HIV, human immunodeficiency virus, the virus leaves the infected cells of the immune system by a process known as budding, where the cell does not undergo lysis and thus is not immediately killed. However, the damage to the cells makes it impossible to function normally, even though the cells remain alive for a period of time. Steps of viral replication. The viral replication is broadly divided into 5 steps, namely, 1. Attachment/adsorption, 2. Penetration and uncoating, 3. Biosynthesis, 4. Assembly, 5. Release. 1. Attachment/adsorption. Since the viruses are obligate parasites, they need to enter the host cell. To enter the host cell, the virus has to first come into contact with a host cell and then has to breach/break the barrier, i.e. the cell membrane in animal cells, and the cell wall in plant cell. Viruses are highly specific and can only attack particular cells of a particular organism, meaning that viruses can affect only certain organisms or tissues. For example, HIV can infect the immune cells of humans, poliovirus attacks the nerve cells, hepatitis B virus, HBV, attacks the liver cells. Plant virus will not affect an animal cell and animal virus will not affect a plant cell. Specificity of the virus is due to affinity/attraction of viral surface proteins to proteins on the surface of the host cell, just like the lock and key mechanism of enzyme and substrate. You have already read in section 1.3.4 that protein coat of the virus particle has capsid, spikes on naked or enveloped virus which helps in attachment with the host membrane. Bacteriophages and animal viruses bind to cellular receptors on the surface of bacteria or animal cell. A well-studied example of the virus-receptor interaction is that of influenza virus. The hemagglutinin protein form a glycoprotein spikes on the surface of influenza virus. This spike binds with sialic acid groups from the glycoproteins of the host cell surface, Fig 2.4. Fig 2.4, Entry of virus into animal cell. Entry of plant viruses into plant cell is difficult as the outer layer of plants are composed of protective layer of waxes and pectin and each cell is surrounded by thick wall of cellulose. To date no plant virus has been found which enters the plant cell with the help of receptors. The plant viruses rely on breach of the integrity of cell wall to enter the host cell. 2. Penetration and uncoating penetration. Once the virus attaches to the cell membrane, it penetrates the host cell either by endocytosis or by fusion of virus envelope, I, endocytosis endo, internal/within, plus cytosis, referring to cell, involves the taking in of matter by a living cell by invagination of its membrane. This is probably the most common mechanism of virus entry into the cell. The enveloped or naked viruses adsorb to the receptor on the host cell surface and are taken into the cell by a process, endocytosis. In this process the plasma membrane of the host cell surrounds the whole naked virion particle forming a vesicle, which enters the cytoplasm. In case of enveloped viruses, the envelope fuses with the vesicle membrane. The viruses then dissolve and rupture the vesicle membrane resulting in the release of the nucleocapsids into the cytoplasm, Fig 2.5. 2. Fusion of the virus envelope. Several viruses are able to directly fuse their envelope with cell plasma membrane without being internalized by endocytosis. After getting attached to the host cell receptor, the envelope of the virion gets adsorbed to the host cell surface and fuses with the plasma membrane of the host cell. The nucleocapsid is then released into the cell cytoplasm, Fig 2.6. Uncoating. Once the nucleocapsid enters the cell cytoplasm of the host cell, viral genome must be separated from its protein coat for replication. This process of release of viral genome is called uncoating. In majority of the viruses, the proteolytic enzymes which break down the proteins, digest the capsid and carry out uncoating. The proteolytic enzymes used for uncoating either are coated by viral genome or is from the host cells. Some viruses like P. coronaviruses do not undergo uncoating. P. coronavirus penetrates the host cell by endocytosis. They directly release their nucleic acid in the host cytoplasm through a membrane pore present in the endocytic vesicle. After uncoating, the released viral nucleic acid takes control over the host cell metabolic machinery. Fig 2.5, Virus penetration of cell membrane of host by endocytosis. Fig 2.6, Penetration of virus into the host cell by fusion of host cell membrane and viral cell envelope. 3. Biosynthesis. After entering the host cytoplasm, the synthesis of viral genome and viral protein starts with help of host cellular machinery. The enzymes of host cells are used for viral genome synthesis and the host cell ribosome and translational machinery is used for protein synthesis. The replication strategy of any virus depends on the nature of its genetic material. We have studied in the previous unit that viruses can be divided into 7 groups, Baltimore classification, on the basis of their genetic material. DNA viruses. Generally, the genomic DNA of DNA viruses is replicated in the nucleus of the host cell. The DNA viruses first enters the host cell's nucleus so that they can replicate and form 2 daughter strands. The central mechanism of DNA synthesis in virus is similar to DNA synthesis in any cell. The synthesis of new DNA strand requires a template, an enzyme which is a host cell enzyme, DNA-dependent DNA polymerase or DNA polymerase. The DNA polymerase uses DNA as template and nucleotides to form a new strand of DNA. The capsid and other viral protein are synthesized in the cytoplasm of host cell. These proteins migrate to the nucleus where it assembles with the newly formed viral DNA to form the nucleocapsid. The nucleocapsid then is transported from the nucleus along the endoplasmic reticulum for its release. Pox virus is an exception as all their components are synthesized in the cytoplasm of the host cell. RNA virus's multiplication of RNA virus occurs in the host cytoplasm and involves formation of mRNA and viral RNA molecules. The enzyme, RNA-dependent RNA polymerase, is required for the synthesis of new RNA. RNA-dependent RNA polymerase is not present in the infected host cell, so are encoded by the virus and are either transported into the cell at the time of infection or are synthesized very soon after the infection has begun. Frequently, the polymerases involved in RNA replication are referred to as replicases to differentiate them from the polymerases involved in transcription. However, both processes are carried out by the same enzyme exhibiting different synthetic activities at different times in the infectious cycle. To study the biosynthesis of genetic material, 3 representative examples of one DNA virus, one RNA virus and one RNA virus, which replicates with help of reverse transcriptase, are being illustrated. Class I double-stranded, DS, DNA viruses, having double strand of DNA, e.g. adenoviruses, herpes viruses, pox viruses, etc. In the nucleus, the DNA of double-stranded, DS, DNA viruses is used for 2 things, one as a template for replication and secondly as template for transcription, i.e. synthesis of mRNA, Fig 2.7. The mRNA formed on entry of the virus into the cell is called early mRNA. These early mRNA molecules are then used as a template for synthesis of proteins, translation. These proteins help the virus in taking over or controlling the host cell's machinery. The double-stranded DNA is replicated using the host cell's DNA polymerase. After DNA replication has occurred, there are a lot of newly made viral daughter DNA which are now available as template for mRNA synthesis. The mRNAs formed after viral DNA replication has occurred are called late mRNAs. The late mRNAs are then translated to form structural proteins. These structural proteins are used for encapsulating double-stranded DNA. The newly replicated dsDNA is then enclosed within the protein capsule. The protein capsule with newly replicated dsDNA then moves to the cytoplasm, to be released from the host cell. Fig 2.7, Replication of double-stranded, dsDNA in Class I viruses. Class 4 contains viruses with ssRNA genomes of the same, positive, sense as mRNA and which can be translated, e.g. P-cornaviruses, togaviruses, etc. In the first step of positive, plus, strand RNA viral replication, viral RNA acts as mRNA. This viral RNA binds to host cells' ribosomes and is translated into early proteins. One of the early proteins synthesized is the viral replicase or RNA-dependent RNA polymerase. The viral replicase uses RNA as a template to make more RNA. Positive, plus, strand RNA serves as templates for synthesis of complementary full-length, strand RNAs. The positive, plus, strand RNA and complementary, strand RNAs together form a double-stranded RNA replicative complex. The newly made negative sense RNA serves as template for synthesis of positive, plus, strand RNAs. These single strands of positive sense RNA are then used as mRNA to make the structural proteins, Fig 2.8. Fig 2.8, Replication of single-stranded, SS, RNA in Class 4 viruses. Class 6 contains viruses that have ssRNA genomes which with help of reverse transcriptase enzyme convert to dsDNA and then form mRNA, e.g. retroviruses. An important element of replication of retroviruses is an enzyme called reverse transcriptase, RT. After infecting a cell, the virus synthesizes viral DNA from viral RNA utilizing the reverse transcriptase. The process by which DNA is copied from an RNA template is known as reverse transcription. Viral genomic RNA serves as template for production of DNA copy. This is produced by an RNA-dependent DNA polymerase, reverse transcriptase, which is part of the virus and enters the host cell with the virus. The double-stranded DNA then migrates to the nucleus and integrates with the host cell genome. This integrated DNA may either remain silent or be transcribed into RNA. The transcribed RNA may be full-length RNA or shorter. The shorter RNAs serve as template for synthesis of accessory and structural proteins. Full-length RNA transcripts are packaged into forming virions. Fig 2.9. Fig 2.9, Replication of Single-Stranded, SS, RNA in Class 6 viruses. The replication strategy of different viruses is different. It is based on the genetic material present in the virus. Fig 2.10 summarizes the different strategies adopted by viruses. Baltimore classification. Fig 2.10, the replication strategy of viruses. Baltimore classification, on the basis of their genetic material depends on the nature of its genetic material. For maturation and assembly. During maturation, assembly of the viral components takes place in the host cell cytoplasm. The assembly process involves collection of all components, virus-encoded proteins and nucleic acid synthesized in the host cell, necessary for formation of mature virus at a particular site. Fig 2.11. For some viruses there is an additional aspect when assembly takes place in the nucleus and proteins synthesized in the cytoplasm have to be moved to the nucleus. Fig 2.11, T4 phage assembly, simplified scheme. The assembly of virus takes place mainly by three mechanisms. I. The various components combine spontaneously to form particles in a process of self-assembly. 2. In some viruses, specific virus-encoded proteins which are not part of virus structure are first removed and then assembly takes place. 3. Virus particle may also be assembled from precursor proteins, proproteins or propeptides, an inactive protein, or peptide, that can be turned into an active form by post-translational modification, which are then modified, usually by proteolytic cleavage, for assembly to take place. The final step of assembly for enveloped virus is the acquisition of the envelope by synthesis of envelope proteins and its incorporation in nuclear, cytoplasmic, or plasma membrane. 5. Release. The viruses are released as a result of lysis of dead cells or by budding/exocytosis from the host cell. I. Cell lysis. For lytic virus, some enveloped and naked virus, the release is a simple process. The infected cell's membrane ruptures, which results in cell lysis and release of the virus. Host cell die because entire host macromolecular synthesis is stopped and only viral nucleic acids and viral proteins are synthesized. As a result, cell death and subsequently lysis occurs, releasing the viruses. Mostly naked virions are released by cell lysis. 2. Budding. Once the nucleocapsid is assembled, some viruses push through the plasma membrane, forcing a portion of the membrane around the virion resulting in an envelope, or fuse with the plasma membrane, releasing the virion. This process is called budding. In this process the host cell is not destroyed during release because the ruptured plasma membrane can be repaired after budding has taken place. Fig 2.12. Fig 2.12, Release of virus from cell. SAQ1. Complete the following flow chart. Answer: 2.3 Replication of bacteriophage, DNA virus, T phage. In E. coli, lytic infections are caused by phages known as the T phages, while lysogenic infections are caused by the phage lambda. On the basis of replication methods, there are two main types of bacteriophages, lytic or virulent type and lysogenic, temperate, or avirulent type. So we can say bacterial cells can undergo one of two types of infections by viruses termed lytic infections and lysogenic, temperate, infections. Lytic phage infects the bacterial cells and multiplies within the infected cell, producing large number of progeny phages. Progeny phage particles are released after the host cell bursts or its lysis occurs at the end of incubation period. This process is termed as Lytic Cycle. Temperate phage undergoes Lysogenic cycle wherein progeny particles are not produced. The phage DNA gets integrated into the bacterial chromosome and is replicated along with the bacterial chromosome. Lysis of host cell does not occur in this case, and the phenomenon of this stable relationship between phage and its host is called lysogeny. However, temperate phages may sometimes spontaneously become virulent and bring about the lysis of the host cell, releasing their progenies. 2.4 The lytic cycle. The bacteriophages of the T-even group, T2, T4 etc., are virulent viruses as they lyse the host cell during lytic cycle of infection. To study the lytic cycle phage T4 replication in E. coli will be used as a model, Fig 2.13. Recall the structure of T4 phage, Section 1.4. Attachment/adsorption. When the phage and bacterial cells randomly collide, the tail fibers of phage base plate gets attached to its specific complementary molecular component called receptor site on the surface of bacterial cells by the formation of weak bonds. This is an irreversible process, which means that attached phages cannot get separated from their host cells. Penetration. After attachment, the tail of phage releases lysozyme, which dissolves portion of the bacterial cell wall, forming a pore. The tail sheath contracts so that the tail core is pushed through the cell wall and comes in contact with bacterial plasma membrane. When it reaches the cell membrane of the bacteria, the protein plug blocking the tail core is removed and DNA is ejected through the core into the cytoplasm, and the capsid remains outside the bacterial wall. Mechanism of phage attachment and nucleic acid penetration are more or less similar in both lytic and lysogenic cycles. Biosynthesis. The viral genome, after entering the host cytoplasm, uses the cell's ribosomes and translation machinery to synthesize new phage proteins, head, tails, and tail fibers. Once viral genome is in the host cell, it takes over the metabolic machinery of the host cell and directs the cell to make only viral nucleic acids and viral proteins. Host DNA gets degraded and the nucleotides as well as the host cell's enzymes are used for synthesizing copies of phage DNA. Phage DNA is transcribed to mRNA, which directs the synthesis of phage enzymes and capsid proteins on the host cell's ribosomes. The newly synthesized enzyme, DNA polymerase, replicates the phage DNA, Fig 2.13. Maturation and assembly. The phage proteins and the copies of viral genome spontaneously self-assemble to form virus particle. Phage head and tails are assembled separately from viral proteins. Each head is then packed with the viral DNA and gets attached to the tail. Tail fibers are then added to form mature infective phages. Release. In the final stage of the lytic multiplication cycle, the lysozyme, encoded by phage genome, causes the lysis of the wall of the host bacterium and the phages are released from the ruptured bacterial shell, Fig 2.13. The time taken from attachment to release is called the burst time. The number of virions synthesized and released from one infected host bacterial cell is called burst size or viral yield. The lytic cycle is called a productive infection, as many virions are produced. Fig 2.13, Lytic Cycle. 2.5 The lysogenic cycle. Lambda phage of the bacteria E. coli is the most widely studied example of lysogeny. The phages whose choice of replication cycle is lysogenic are called lysogenic phage, temperate phage, and the bacteria in which it occurs is called a lysogenic cell. In the lysogenic cycle, the phage gets adsorbed to the bacterial cell surface and penetrates its linear DNA genome into the cell. In the host cell cytoplasm, the phage DNA forms a circle, circularizes, and gets inserted into the circular DNA chromosome of bacterium. The viral DNA integrated with bacterial DNA is called prophage, and the combination of the bacterium and temperate phage is called a lysogen. Two genes present on prophage produce two repressor proteins and repress the transcription of all other prophage gene. The prophage gets replicated along with the bacterial chromosomes during reproduction and remains latent in the bacterial progenies. As the prophage does not lyse the bacterial cell, the infection is referred to as latent infection. When the bacterial cell becomes stressed due to UV radiations, presence of chemicals, the prophage excises itself from the bacterial chromosome and is released in the cytoplasm. This dissociation is called induction. After induction, lytic cycle is initiated, lysis of the bacterial cell occurs and new phages are released, Fig 2.14. There are three implications of lysogeny. 1. the lysogenic cell becomes immune to infection by another phage of same type, a process called lysogenic conversion. However, the lysogen can be infected by other types of temperate or virulent phages. Secondly, the lysogenic cells may exhibit different properties than the non-infected host cell. For example, the bacteria Corynebacterium diphtheriae, that causes diphtheria, and Clostridium botulinum, the causative agent of botulinum, contain prophages, each of which has a gene which codes for the production of a toxin. The disease-producing properties of these bacteria are related to these toxins, as without the prophages these bacteria do not cause diseases. Thirdly, lysogeny enables the specialized transduction. In this process, when the prophage is excised from host bacterial chromosomes during its lytic cycle, the adjacent genes of bacterial chromosome from its either side may remain attached to this phage DNA. As a result, the phage particle packages the bacterial DNA segment along with its own DNA in its capsid and carries the same to another bacterial cell it infects. Fig 2.14, Lysogenic Cycle. 2.6 Replication of Anavirus Tmv. Recall the structure of Tmv, section 1.5. The Tmv enters the cell through cell sap via abrasions/wounds on the host cell surface, as it does not have any specialized organ for attachment as observed in bacteriophage. Once the virus enters the plant cell, the viral genome i.e. plus ssRNA is released into the cell after uncoating takes place. Uncoating occurs by dissolution of protein coat, capsid, by the host cell enzymes. They, plus ssRNA acts as mRNA, and replicase gene is translated with help of host cell ribosomes. The newly formed replicase enzyme is used to replicate the viral genome. Plus, ssRNA acts as a template and replicates with help of replicase to form a complementary ssRNA. They, plus ssRNA and complementary ssRNA form a plus or minus double-stranded RNA called the replicative intermediate, RI. The complementary ssRNA of replicative intermediate is used as template for synthesis of more plus ssRNA. This plus ssRNA is used as mRNA and translated with help of host cell machinery to form capsid proteins. Once enough capsid proteins are produced at an optimal level, plus ssRNA formed is used as viral genome. Fig 2.15. Fig 2.15, Replication of Tobacco Mosaic Virus. The TMV viral genome, plus ssRNA and capsid protein combine spontaneously to form particles in a process of self-assembly. SAQ2 Fill in the blanks. I. The virus penetrates the host cell by two main mechanisms, fusing of envelope and DACE. Answer Endocytosis. 2. The process in which various viral components combine spontaneously to form particles is called DACE. Answer Assembly. 3. The bacteriophages of the T4 lyse the host cell during DACE cycle of infection. Answer Lytic. 4. A process by which viral genome is pushed out into the cytoplasm by break of capsid is called DACE. Answer Uncoating. 2.7 Summary. In this unit you have learnt that Z virus particle, virion, cannot replicate outside the host as it lacks the desired cellular machinery that are required to reproduce. Z viral replication results in many progeny, and these progeny leave the host cell to infect other cells in the organism or spread the infection. Z virus replicate by a 5-step replication: attachment, penetration and uncoating, biosynthesis, assembly and release. Z. The virus-host cell receptor interaction helps in the attachment of virus to the host. Plant viruses enter plant cells by breach in plant cell wall. Z. The virus penetrates the host cell by two main mechanisms, endocytosis and fusion of envelope. Uncoating is a process by which viral genome is pushed out into the cytoplasm by break of capsid. Z. The strategy for biosynthesis of genetic material of viruses depends on the nature of its genetic material, DNA or RNA. Z. The assembly of virus takes place mainly by three mechanisms: by a process of self-assembly, specific virus-encoded proteins are first removed, not part of virus structure, and then assembled from precursor proteins, which are then modified, usually by proteolytic cleavage, to form the infectious virion. Z. All viruses escape the cells by one or two mechanisms, budding and cell lysis. Z. Bacterial cells can undergo one of two types of infections by viruses termed lytic infections and lysogenic, temperate, infections. 2.8 Terminal questions 1. With help of diagram discuss viral replication, general account. Answer section 2.22. Differentiate between lytic and lysogenic viral replication. Answer section 2.4 and 2.53. Give in detail the biosynthesis of DNA viruses. Answer section 2.2, 3, biosynthesis 4. Discuss the replication of Tmv. Answer section 2.6 Glossary Assembly, the gathering and replication of viruses within a cell by using the metabolism of the host organism. Attachment, the condition where the capsid proteins of the virus bind to certain receptors of the host organism. Bacteriophage, a virus that must infect bacteria in order to reproduce itself. Burst size, the burst size of a phage is the average number of new phage produced by a single infected bacterium during the lytic growth cycle. This number varies widely for well-studied phages, from only a few to over 100. DNA polymerase, one of a class of enzymes of complex structure that catalyze the synthesis of a new strand of DNA complementary to the template strand in a primer-dependent reaction. dsRNA, double-stranded RNA. Endocytosis, the process of incorporation of viruses or large molecules into a cell by formation of a specific vesicle at the cell surface that engulfs the material and transports it into the cell's interior. Endoplasmic reticulum, the complex membrane system in eukaryotic cells that is continuous with the nucleus. It is the site of lipid and membrane synthesis as well as the synthesis of proteins destined to be secreted or remain membrane associated in the cell. Gene expression, an activity where information from a gene is made into functional gene material. Incubation period, the time between initial infection and the onset of notable symptoms of a disease. Genome replication, the reproduction of genetic material, particularly that in the structure of DNA. Latent infection, a viral infection that exists in dormancy and does not exhibit symptoms. Isogeny, ability of certain bacteriophages, notably, bacteriophage L, to integrate its genome into that of the host bacteria and remain associated as a genetic passenger as the bacteria replicate. Maturation, the phase during replication at which a virus becomes infectious. Penetration, the process of the virus entering the cell of the host organism, causing infection. Plaque, a defined area of cell destruction resulting from virus infection in vitro. Plaque-forming unit, PFU, a unit of infectious virus determined by the ability of the virus to form a plaque or area of lysed cells on a lawn of susceptible cells. Prophage, prophage is a bacterial virus the genome of which is residing in a bacterial host in a passive state, not expressing many viral functions or producing progeny virions. It may be incorporated into the bacterial DNA or it may be a separately replicated molecule within the bacterial cytoplasm. In some cases, toxin genes present in prophage genomes have gene expression signals that allow their independent function when the viral genome is otherwise passive. Receptor, a specific type of molecule found on a cell membrane that a virus is able to attach to. Release, the process of the death of a host cell that discharges a virus. Reverse transcriptase, an enzyme, originally discovered in retroviruses, that uses single-stranded RNA as a template for the synthesis of a cDNA sequence, can also use DNA as a template, and can degrade RNA from a DNA-RNA hybrid. This latter activity is termed RNase activity. Temperate phage. Temperate phage is capable of establishing a lysogenic state in a susceptible bacterial host. This is a quiescent state in which virus replication does not occur. If this option is not available to the phage is referred to as lytic. Transcription, the enzymatic synthesis of RNA from either a complementary DNA template. Uncoating, a condition when the protein capsid of the virus is unsheathed due to enzymes of the cells of the host organism.